Okay, so as we all know, that the bacterial resistance to all major classes of antibiotics is increasing, and this is one of the most severe medical and public health problems we are facing today. So the initial question for our project is, can we get reversion back to sensitivity? And if not, how could we optimize the strategy for antibiotic resistance control? We hope to answer these questions by modeling the evolution of drug resistance at the population level. And before I start to talk about more details of our model, we need to know how resistance evolves and why is it so hard to eradicate those resistant pathogens. Here's one explanation. And this is the fitness landscape of bacterial strains under different environmental conditions. And the term fitness of a strain here can be defined as its growth rate in a given environment. And the resistant mutant comes with a fitness cost to the sensitive strain in the absence of antibiotics. And one example about this could be bacterial enzymes that modify antibiotic often leads to metabolic cost and then leads to reduced growth rate. People used to think such cost of resistance would raise the possibility that prudent use of drugs may slow or reverse the rise of resistance. However, later people have observed such cost can be compensated or even eliminated by a subsequent mutation, so we call it compensatory mutation or compensated resistant type. In the absence of antibiotics, due to these relative high and low fitnesses of different bacterial strains, the selective pressure pushes the bacteria to mutate from resistant type to either sensitive or compensated resistant type. While in the presence of the antibiotics, the selective pressure pushes the bacteria to evolve from sensitive to resistant, then to the compensated resistant type. As the environment alternates between these two scenarios, our bacterial population may be trapped gradually at this end, where reversion becomes hard. And this phenomena was described by Backstrom and Feldgarden as the evolutionary lobster trap. And given this crucial impact of compensatory mutation in the evolution of drug resistance, it is very important to study whether and how this trap can be escaped. And Tanaka and Benken both have investigated the dynamics and point out crucial factors for reversion at the within host level. And we would wonder what happened at the population level. And here's the structure of our model, which is built on a classic epidemiological model. And the dynamics of this model can be described using a system of differential equations, but don't worry about those model details here today. So the only thing I want to say about this picture is we are able to use this model to analyze the disease dynamics mathematically. And a key assumption about this model is about the process of susceptibility testing. We assume susceptible individuals can be infected with 3 types of the strains: sensitive, resistant, or compensated resistant to a first-line therapy, which is usually recommended on the basis of clinical evidence for its efficacy. As traditionally, a proportion F of our infected individuals are directly treated with this first-line drug. And this may lead to a consequence that patients infected with R and C strains would not be treated as effectively as the sensitive case. And this would also give extra time or chance for the resistant bacteria to evolve to become compensated resistant. To improve this situation, we model a scenario where each new detected cases can be tested for resistance. The parameter rho here is the actual proportion of detected cases being tested for susceptibility to the first-line therapy before the treatment. If resistance were found, an alternative second-line therapy would be applied. And this drug may previously be preserved from the community because it is more toxic or it is more expensive than the first-line therapy. And before the rise of resistance to this second-line therapy, we should now be able to treat R and C cases as effectively as the sensitive case. And based on these assumptions, here is what we found. And this phase diagram shows the final equilibrium status of a disease, which initially starts with patients infected with sensitive strands only. And we have 3 types of the outcomes according to different treatment proportion F on the horizontal axis and testing proportion rho on the treatment proportion F on the vertical axis and testing proportion rho on the horizontal axis. And for example, if we are at somewhere here where F is small and rho equals 0, that means a very low proportion of the infected individuals would be treated and none of them would be tested for resistance. And if we are at somewhere here instead, that means approximately half of the patients would get treated and all of them would be tested for susceptibility to the first-line therapy before the treatment. So what does this diagram tell us? In the blue region, we end up with sensitive epidemics in the population, and that's when a relatively low proportion of the infected individuals are treated and a fair proportion of them are being tested for resistance. And if we treat more patients but do not test them enough, we may enter this red zone where compensated resistant pathogen reaches fixation in the population at equilibrium. And remember that question we asked at the beginning of this talk, can we get reversion back to sensitivity at the population level? The answer is probably no here in this red region. And this would be the disaster situation for antibiotic resistance control. And unfortunately, for many bacterial infections we see in the hospital today, since we don't do any susceptibility testing for patients before the treatment, we are currently at somewhere along this vertical line. And fortunately, disease eradication can be achieved at a high treatment proportion and a moderate to high testing proportion. And to briefly conclude, susceptibility testing and an alternative second-line therapy would largely affect the evolution of drug resistance at the population level. And from this model, we can get this good qualitative understanding in the control of antibiotic resistance, particularly through the interaction between treatment and testing proportions. However, high treatment and high testing proportions mean that the second-line therapy would be used at a high rate too. So this would challenge one of our assumptions: resistance does not evolve to this second-line drug. So to solve this problem and find out what's actually happening there at the top right corner, we have extended our model so that the evolution of drug resistance to the second-line therapy is included. Pathogens are now given by 2 symbols rather than 1. The resistance status to first and second-line therapy at the first and second positions respectively. For example, strains sensitive to both drugs are labeled SS, and strains resistant to both drugs are RC or CC, depending on whether cost compensation has occurred at the first position. And we will now have more types of pathogens in total, and we follow the same testing procedure as before. And here's what we've got. So comparing the outcome of the extended model with our previous results on the left, we find the disease is much harder to eradicate because under the same parameter values here, because the doubly compensated resistant pathogen would dominate the population in most of our parameter space. And a critical boundary of interest here would be the one that separates disease eradication from the fixation of CC strain. And it is affected by the strengths of resistance to both first and second line therapies. For example, the left diagram here has a larger eradication zone because the CC strain in this case are less resistant to both drugs than those in the right diagram. And our paper has been recently accepted by academics, and the model can be extended in various directions. For example, it could be made epidemiologically richer by including recovery with immune memory and other infection-specific features. And another missing element here would be bacterial recombination, which also facilitates the evolution of drug resistance, of compensatory mutation. So the effect of recombination in a multi-drug system would be interesting to investigate further. And when I gave a talk about this project in the Genetic Society of Australasia conference in July this year, one clinical doctor named Peter Tsai from Taiwan showed great interest in applying our theory to the resistance data collected from their surveillance program. And in the current literature, we've seen many reports on antibiotic resistance surveillance from different regions and countries across the world. But as Peter said, people don't often analyze this data thoroughly due to a lack of theoretical understandings. Therefore, in future work, we hope to collaborate with clinical researchers who will provide patient data to increase our confidence in the estimates of our model parameters. And another crucial aspect which may increase the effectiveness of controlling resistance through this model is to improve the accuracy and the efficiency of susceptibility testing. And each of these points here would require a large amount of time input and as well as fundings from medical and public health organizations. And if we could ultimately fit real data to our theoretical model, the community would greatly benefit from optimized strategy for antibiotic resistance control. And thank you.